It gives me great pleasure to introduce Martina Summer Kutmon from Maastricht Center of System Biology in the Department of Bioinformatics, which is in the past also where I worked. So I know Tina from past work. And maybe to start the introduction, there is an anecdote I would like to share that relates to SWAT4HCLS as well, because when we were preparing the introduction today, we realized that the first presentation of the WikiPathways RDF work actually happened at SWAT4HCLS in 2012 in Paris. But before that, we also we went to Heidelberg also in 2012, where we met with others in a cafe in Heidelberg, where we generated the ontologies that later became the pillars of WikiPathways RDF. And why I'm saying this, because I often think about this story again, because we did 80% of the work to build the ontologies and the RDF during that breakfast session. But it took us years to finish the last 20%. And it's a pattern we often see since. So all you need is actually to get semantic web data, is to get an expert like Tina in a cafe in Heidelberg and try to design ontologies in RDF. And with this anecdote, I would like to give the floor to Tina, and looking forward to your talk, Tina. You have the floor.
Thank you very much, Andra, for this very nice introduction. It's a great pleasure to be here today to join this conference and to listen to the diverse topics and inspiring talks so far. Today I will talk about WikiPathways, and Andra already mentioned it. There's a semantic web version for that, an RDF version that I will also be mentioning today. I started my PhD back in 2010, and I joined this WikiPathways team. And so my whole life started to turn around molecular mechanisms and understanding them and mapping them out and developing the tools for that to understand how the cells function. And so besides doing this curation myself, the focus was really on the development of methods and tools to make this as easy as possible for curators to create these pathway models and to then allow us to analyze and visualize the data with these models. And a couple of years back, I started to focus on understanding the interplay between metabolism and immune system, especially in chronic diseases and cancers. But then COVID hit, and so the research changes. And so also we started to do a lot more research in COVID, trying to better understand what happens on a molecular level when there is a virus infection. Can we create these pathway maps and so on? We were able to get some funding, and so there were a lot of developments, not only on the curation side, but also on the technical and the tools side, because we realized there were certain features that were really essential for such a fastly developing research area. And so today I'll give a little bit of an introduction of WikiPathways and then show some of the developments and applications that we're working on. So as one of the WikiPathways architects, I like to start with an acknowledgements page, because this is a real community project. So the work that I'll present is really done by a lot of different people. So Alex Pico and I, so Alex is from the Gladstone Institute in San Francisco. We lead a team of core developers and admin curators, but a lot of the content and many of the features that come actually from contributors and curators from around the world. And so WikiPathways itself and all the associated tools are developed under an open source license and available on GitHub, and really we encourage this kind of open spirit that the project has. So many of you have probably heard of WikiPathways, but I'll give a short introduction of the fundamental concepts of the resource. So when we talk about pathways, this might be the first one that we think of. It's a well-known early example of curated pathway diagrams. These are all the metabolic reactions that we knew back then, with the TCA cycle in the center, describing this metabolic map that's going on in our cells. These were distributed as wall-sized posters, and they look hopelessly complex and complicated, but they were actually quite simple and relatively stable. That's not so true nowadays anymore, because we gather more and more information all the time. So nowadays, pathways are very focused, but they become more detailed and more complicated. They come in many different flavors. We have high throughput measurements, disease-specific ones, drug actions, and so on. And we can integrate all that information in our pathway diagrams. We can find them, therefore, in publications, in presentations, in posters, everywhere. And the utility to biologists and researchers as kind of models is obvious, because we have a very clear understanding when we look at such a diagram, oh, this is what's going on, and we can discuss and share these concepts. What we are interested in is, especially when we model these diagrams properly, so if we make them more than just images, we make them machine readable, then they become immensely useful for computation analysis. And that's really important because we produce so much large-scale experimental data nowadays that it's essential to have tools that facilitate the interpretation and analysis of these data sets. That means that these pathways nowadays are more than just images. They have the visual component, but they're machine-readable models that have a lot more semantic meaning underlying the visual representation. As an example, I like to give this example. So we have this static image, which was published in 2016 in a Cell paper, where we see a nice signaling pathway. I'm just using the laser pointer here. We see here there's a membrane protein, and then there's a whole set of signaling cascades happening, some changing the transcription of certain genes. What we also see is that there's 3 different types of data visualized on the pathway. These are indicated with these arrows. They have 3 types of data: mRNA abundance, protein abundance, and phosphorylation, so phosphoprotein abundance. And a new data set would then require a manual change of each of these arrows next to the data nodes. If the pathway would be created in a machine-readable format, like we've done here, a colleague of mine from San Francisco did this, and submitted it to WikiPathways, we can now show any data set on this pathway in a quick and an efficient way. And the same data could also be visualized on other pathways without additional work. So what we aim for is that authors of pathway figures like this would additionally provide such a pathway model before its publication. That would be the ideal world, because then the publication would have a reference to the machine-readable format, which becomes part of the publication and directly increases reproducibility and reusability of the knowledge. In pathways, we often try to integrate a lot of information about the process that we know, but we also know that this will change over time, especially if we think about COVID. Initially, we knew a lot from SARS-CoV-1, but then more and more details came in, and we needed to constantly adapt the models that we created. So the idea of WikiPathways was launched in 2008 as an experiment for community-based curation of biological pathways. This morning in the first keynote, we were talking about the increase on publications that we saw in COVID, right? So even in 2008, we thought that there's too much knowledge and too much data out there, and it's already difficult to keep knowledge up to date, accessible, and integrated. And now it's probably a lot worse. And the idea of WikiPathways is that we take advantage of the direct participation by the greater portion of the community. In this case, we talk about the experts, the expert researchers themselves. And that's the concept of crowdsourcing. So let's all work together. Let's get the experts involved. And so what happened is that Alex Pico and the team in Maastricht, when I wasn't there yet, so Thomas Kelder and Martijn van Eersel, they started the WikiPathways project. It's like a wiki, like Wikipedia, but they ripped out the text editor and replaced it with a pathway editor. So they wanted to have this visual component very clearly. And what that meant is that we now have a collection and curation of knowledge about molecular mechanisms that is community curated, which means everybody can find a pathway, contribute new pathways, or add information to an existing pathway. In the spirit of open science, all pathway content on WikiPathways is released under a CC0 license, which also increases reusability of the content. Just because the content is there, people can continuously work on it and improve it. This provides a platform where new findings can be added immediately, ideally by the experts themselves, right? So if you find a new inhibitor or a protein that activates another protein, you can add it to the diagram directly. Discussions about content are strongly encouraged. We want research to exchange ideas, to discuss the pathways, to add information to other people's pathway, and so on. And we have collaborations with other manually curated pathway databases like Reactome or NetPath to provide feedback from the community. We use the concepts of quality tags. These are kind of annotations which give information about the curation status of a pathway and the context of the pathway, to already upload pathways even though they're not ready for analysis yet. So they might still be under development or hypothetical pathways. We simply tag them as such, and then they are not included in the downloadable analysis collections. That also allows us to add annotations using the cell type and disease ontology to say this pathway is relevant for a specific disease purpose or in a specific cell type. So we also allow these kind of specific versions of pathways. On WikiPathways itself, every pathway has its own pathway page with the title, author list, and clickable diagram. So you see this very nicely here. The first author is always the original submitter of the pathway, and afterwards, based on the number of contributions, the authors are listed there. When you click on the diagram on the element, each element is annotated with an identifier. And if you click on the element, cross-references are provided by BridgeDB. So the curator might have selected the NCBI or former Entrechin identifier when annotating, when creating the model. But BridgeDB then provides cross-references to Ensemble, to gene ontology, to different microarray platforms, and so on. Additionally, you can also find these mentioned quality tags and the ontology tags on the pathway page. These provide insights in the curation status, so how far is the curation level, but also the biological context. So in which pathway ontology term are we talking about, for example, or which disease is relevant here. In the pathway, each element, so the nodes and the interactions, can be annotated with publication references. And that provides this bibliography that basically shows you all the references, all the literature, all the material that has been used to create this one pathway model. Every pathway page also has a discussion page. As mentioned, we really want people to interact and discuss the pathways. And you can download the pathways in different formats, from machine-readable formats for analysis to different types of image formats. And just a heads up, we're working very hard on a new website interface that's hopefully coming soon with some nice, cool browse and search features. Additionally to the larger species-specific pathway collections, we also enable communities to create and maintain their own focused collections of interest. We call them community portals. So we currently have over 20 portals for different communities, where they curate and select pathways that are relevant for their own research. That might be going from the plant portal to rare diseases to COVID-19. A lot of different projects working on this. I'll talk a little bit more about the COVID-19 portal later. Overall, we have over 780 contributors so far, with over 3,000 pathways for over 20 different species. Some statistics from last year, so we have continuous over around 15,000 visitors a month, but there's also a lot of usage through the automatic programmatic access through the REST web service. Additionally, we really focus on disseminating our content in other resources to reach a wider international audience as well. This is a figure that we published in the NAR 2021 database issue, which shows a nice growth, not just in genes and proteins and interactions on the right side, or pathways in the middle, but also on contributors and revisions. So there's a continuous change of the platform visible over the last 4 years in this case. So in the title of my talk today was also very much linked to this last NAR database issue, where we focused on connecting these communities. So on one hand, we talk about the curation communities on the left side in this graphical abstract, all the different communities and researchers that contribute their knowledge to WikiPathways. And then on the other hand, we have communities that use the pathways for analysis, for data integration, for understanding what's going on in their data. And so today I'll show a little bit of both of these aspects. When you want to access data from WikiPathways, you can do this from the website, but you can also access it programmatically. That's very relevant. One is the REST API, as I mentioned. The other one is the RDF version. So we have a semantic web version that I'll explain a little bit more as well. If you're doing processing in R, you can also use the R WikiPathways app, or use the Cytoscape app if you're doing more network analysis. Now about the wider dissemination. Index is the network data exchange platform where we have all our human content integrated in this platform and further share it with the community. And the same we're doing in Wikidata, where we have the metadata about the pathways included there. The TMT files is a nice way for gene set enrichment analysis. That was the one that we created last, and it's now integrated in MSigDB as well. So if you're doing kind of enrichment analysis, the data is there now available as well. So how do we actually store these pathways? And for that, I'm going to very shortly explain the graphical pathway markup language, GPML. That's the file format that we use in WikiPathways. That's how we store and share the pathways. And I'll show a little bit about how we create these models and then the storage of it. So the pathway editor that we use is a standalone application called PathVisio. As you see in the screenshot, we have this panel on the right. It's a little bit like PowerPoint where you just click and drag and basically create your image. Then by double clicking on the elements, you can add further information about the data nodes, including identifiers, comments, literature, and so on. We're actually working very hard as part of the COVID-19 grant on a PathVisio 4, where we have a lot of new features that will facilitate curation as well and standardization of the models. And about standardization, I also wanted to shortly mention this 10 simple rules paper that we published last year as part of the community efforts. But we learned a lot about how researchers submit their pathway models to our database. And so we try to have a couple of rules, guidelines that help you. The first, some of them are linked to detail. So sometimes you might have more detail, sometimes you might have less detail, depending on the context of the pathway. We talk about colors, use of colors and the difficulty when you use colors for data visualization later on. And we talk about keeping your pathway alive in a way. So basically it's growing and it's changing over time. But there's also several rules that talk about semantics, the importance for semantics when we talk about pathway curation. It's important to use standardized naming conventions and identifiers, that a protein is annotated with a UniProt identifier, that a metabolite might be annotated with a CABI identifier, and so on. There should always be a title, a meaningful title that is easy to understand and covers what the pathway describes, as well as a description and ontology terms that give you context in a machine-readable format. You can kind of categorize the pathways a little bit this way. Additionally, as much standardization as possible in terms of interaction types. There we're improving also with the next PathVisio version a lot. And importantly, disseminate your pathway models in other resources, in other formats, to make sure that as many people can use it for their own purposes that they might need. When we draw a pathway, we draw it interactively. So you click and drag and create this catalyst and a substrate and a product, and you connect them. Underlying, you create this GPML file. This is the GPML 2013A version. That's the schema that we use. And if I look at this example, the catalyst here, I have semantic information. So I know that it's a gene product. I know that it has a UniProt identifier, but I also have a lot of graphical information. I know exactly how big the box needs to be, where it is positioned, and so on. So you have combination of graphical elements and semantics that you can get out of it. The same with the interactions, right? So we have the interaction says to which elements does it connect to? And then it also says what type of interaction is it. In this case, it's a MIM catalysis. MIM is for molecular interaction map. That's the type of interaction we're using here. So the 2013A schema combines semantics with graphical information. So the focus is really to be able to exactly reproduce the image by keeping as much of the semantics there. And the annotations of data nodes and interactions happens with these XREFs, so these identifiers and data sources. And adding literature references is really important for provenance. Where does the information come from? The other annotations that you might want to add so far in 2013A are unstructured. So sometimes people add information about phosphorylation states or other elements. And with the TPML 2021, which we already released and we're working on updating all the tools with now, we provide a new way to add these annotations in a structured way. And I'll give you a couple of examples. So Finterly is a scientific programmer at the department, and she worked on the PASVC4 and on this TPML 2021. So if we can now use ontologies to add structured annotations to the different elements in the pathway, we can get a lot more automatic machine-readable semantics with the pathway model. As an example, if I have this pathway as an example here, I have protein A and B. A gets transported from the cytosol into the mitochondria, and there it inhibits protein B. But there's also a virus protein that might interact or bind to protein A. So we can, for example, say that the mitochondria, the protein A and B are located in the mitochondria. We can give them a subcellular location using a Go ontology term, which is for subcellular location mitochondria. The same for the other two. We can say they're cytoplasm, and the TPML 2021 will then allow that. Additionally, we could say that this SARS-CoV-2 element here, this virus protein, is actually from the taxonomy of SARS-CoV-2. So we can have multi-species pathways and identify which elements come from which species. And lastly, and that's something that we realized with COVID-19 a lot, is the importance of annotating evidences. So very often, the experiment was actually maybe done in mouse, but we put the information in a human pathway, right? The same happened initially with SARS-CoV-1 a lot. So we knew SARS-CoV-1 worked this way. So we assumed SARS-CoV-2 works similar. And a lot of the initial pathway diagrams were actually inferred from the other virus, the earlier virus variant. So annotating them with the evidence and conclusions ontology terms like this, for example, that it was an homology evidence-based reconstruction, is really, really important to make sure that the user, the researcher that uses the pathway model, knows what is the evidence for these interactions in this pathway model. And that allows us a lot more deeper and semantic more meaningful queries to perform as well on the pathway models. So this enables the structured and detailed annotations of the mechanisms and context of the pathways. And some of these can actually be automated. So we're also thinking about that. So if we know the cellular location with the graphical elements, maybe we can automate some of these things. But a lot of them things depend on the curator's knowledge that they include these annotations when they curate their pathways. So when we have this TPML files, can we also create this kind of semantic web representation of the pathway content? And this is what Andra referred to our nice breakfast lunch meeting in Heidelberg, where we were like, okay, how can we represent this TPML file in RDF to be useful when we do kind of this linked data approaches? Obviously, the idea is that we want to go over the separation of each TPML file separately, each pathway individually. We know that these pathways are not separated, right? They're connected to each other. There's crosstalk between the pathways and so on. So we can make one large kind of collection of triples that describes all the pathways, and the gene product might be part of multiple pathways, and that is then known in the RDF. The way we approached this was with 3 levels. So we have the TPML file, Then we have the TPML vocabulary, and we have the WikiPathways vocabulary. So that's how we call it. From the TPML vocabulary, we can always go back to the TPML files. We can completely reproduce the TPML file. The WikiPathways vocabulary really extracts the semantics of the pathway model as a whole. So if we look at this example, I just picked this interaction out of a pathway model. We see in the TPML file, we have the combination of graphical, so mainly graphical information about the font size and the font color and the height of the box and things like this, with the identifiers and maybe some additional semantic information. In the TPML vocabulary, we actually retain all the graphical and annotation details. So we have a TPML predicate of font weight, and then we can have the font weight defined there. So each element in each pathway has a kind of representation in the TPML vocabulary that allows us to go back to the TPML file if needed. And the WikiPathways vocabulary then infers the biological meaning. So it doesn't care anymore how big the box is. It doesn't care about where the box is located. It cares about what is the biological element. And for that, we use identifiers.org identifiers. It cares about the identifiers, the data sources, what is it about, and things like this. Using BridgeDB, we are also enabling some identifier mapping already in these kind of approaches. When you go to the SPARQL endpoint of WikiPathways, Marvin and Denise, they're currently working on, so Andra and I might have kicked it off initially with the vocabulary. Andra implemented a lot of the initial RDF. Then Ryan is a former PhD student who took over that a little bit, and now Denise and Marvin and Amar are continuing that work and are further developing the RDF. And they developed the Snorkel interface, which actually provides a whole set of example queries, including some federated queries with other resources like Nextprod, where you can run through and try out things. Obviously, if we change all the pathways to TPML 2021 and we allow more depth in the semantic annotations of the elements, then we also will update the RDF schema accordingly. So that's a work in progress. Now, a little bit about applications and what we're actually doing. Many of you have probably heard of the Disease Map project by now, the COVID-19 Disease Map project. I think it was mentioned in one of the questions earlier. When COVID hit, we started to create pathways quite quickly, and we were not the only ones. So there were hundreds of researchers that were trying to understand the molecular mechanisms. And this formed this kind of community of pathway curators, which is the COVID-19 Disease Map project. And WikiPathways is one of the teams that contributes models to this project. And this is the current overview of the disease map. And you see the different steps of the virus infection, where the virus enters the cell over ACE2, and then there are several pathways happening. So each of these red boxes has a pathway model underlying, or multiple sometimes. And they can come from WikiPathways, they can come from Reactome, and they can come from other pathway curators that use different tools and different methods. And so it's kind of a collection of all of these approaches together. And it's a very successful and fulfilling project because there's a lot of interactions and exchange with a lot of researchers with the same aims. So it's a very nice initiative. On WikiPathways, we developed this COVID-19 community portal where we currently have 38 molecular pathways. And the earliest model actually started to be submitted in February 2020 already. So this portal was set up already in March because we realized this is going to be something that's going to be very active over the next few, I don't know if we immediately expected 2 years, but at least for the next times. And we were able to get funding from SONMV to work on the pathway curation, so curate pathways, but also develop workflows and tools that are relevant. And I'll show a little bit about the kind of approaches that we're using, always focusing on reproducible, automated analysis workflows to make it really easy for others to reuse and reproduce. The example dataset that I will show is an RNA sequencing dataset from the primary human lung epithelium cell. That's the cells in the lung that can get infected by the SARS-CoV-2 virus and are very often affected. They were either mock treated, so they were the controls, or they were infected with the virus. And we basically look at what is happening on a molecular level in these cells. So initially we saw that there's a lot of upregulation, stronger upregulation of certain genes, and also some downregulation. And as expected, there's a lot of immune response going on, right? So we have the highest upregulated, or most significant and highest upregulated gene is a cytokine, so CCL20. There's also some apolipid proteins that those react to inflammation. But we also see some, for example, S100 calcium binding protein A8, which is now a proposed target in their clinical trials. So we see there's a lot of changes happening on a transcriptomics level. We then did this pathway analysis. So we used all the pathways that we have in WikiPathways, and we do some statistical tests to check which ones are enriched for genes that are affected in COVID infected patients. And we can visualize these in the pathway diagrams, which is the advantage of having these automated models additional to just knowing which genes are involved in the process. We can actually look at them and see what's happening, like we do here in the type 1 interferon pathway, for example. But we can do more. We can actually integrate these pathways in networks and add some drug target information, because obviously we're interested in maybe interesting drug candidates or targeting treatment options. A lot of differentially expressed genes are not yet in the pathways or are not in the differentially expressed pathways. So for those, we will need to do additional analysis if needed. So if we add the drug information, we get over 444 drugs that target genes in that pathway, in those pathways that are affected. But they're not independent and they all overlap. And that's also what we see here. So when we looked at these 444 drugs, 245 were actually FDA approved and some others were still under investigation. 92 genes were actually targeted. So there were very different processes affected as well. And when we looked more details, so for example, here we look at the TNF pathway. There are certain drug candidates that you might have heard, I'm sure as well, like chloroquine or ibuprofen and things like this. There's also the tocilizumab, which is currently, it's an experimental unapproved treatment of COVID-19, where there's trials ongoing. That's an interleukin-6 receptor inhibitor that's known to treat the cytokine release syndrome. So the cytokine storm is something that causes these severe cases or is associated with the severe cases of COVID. Something when we talk about curation is knowing what we don't have in our collection yet, right? So we don't have all the information in our pathways. So how can we find out which elements we don't know? And so what we did, we integrate another data resource. In this case, we used the string database for protein-protein interactions, and we connected all the differentially expressed genes. And we see some nice clusters here. For example, here, but also here, unconnected clusters that are tightly connected and share a similar expression change after infection. And what we did is we extended this with pathway information. So all the green nodes now are pathways in our COVID-19 disease map. So they are from Reactome, WikiPathways, and other models. And we see which genes are already present in some pathways and which ones not. And this allows us to identify new curation focus. For example, only the genes with a yellow border are in a COVID-19 pathway, right? But there's some genes here that are tightly connected. Let me just say all the yellow ones are in a pathway, but what about these three? These are very tightly connected to these others. Maybe they should also be part of these pathways, and can we extend the current pathways with adding more information? And we're doing this obviously with multiple different data sets, and the more often a gene shows up in such a analysis, the higher the curation priority goes. When we talk about pathway quality, right, we also want to assess the quality of these pathways. And one way is the quality tags, right? I can say this is still under development or there's missing identifiers or things like this. and some are automatic. And Egon Wellichhagen is doing a lot of work on this kind of computer-assisted curation. So we saw that people repeatedly make the same mistakes, and so do computers. So you see the same little things that they do wrong. Some things are hard to detect if an identifier is outdated, or if there's a typo, or there's an inconsistency, things like this. And so these computer-assisted curation reports that are generated, they provide an overview of the curation status of a pathway. Initially, we did this it was run in Jenkins, where all based on the RDF. So we used the GPML RDF and the WikiPathways RDF. And then there were different tests for metabolites, tests for genes. Are there outdated identifiers? Are there identifiers that don't match the pattern that it should? Things like this. You then got this kind of result where it said, oh, all the tests are green, or all the tests are red, or 3 are red, and so on. And you could check what actually caused the problem. Now, recently, Egon has worked a lot on improving the output so that the visual representation of this result as part of the COVID-19 and the EpiLipidNet projects, where you now actually see a lot more information in such a report. So it generates the WikiPathways RDF file and the GPML RDF file, but it also generates an SBML file, so Systems Biology Markup Language file, and so on. For each test, you can see, was it okay or not? Were there any errors? How many errors? And further down, you can check what the errors were exactly. These are all markdown files, which are generated on GitHub. So the whole automatic assessment of the COVID-19 pathways, actually at the moment, they're done using GitHub Actions, and they produce such an MD file for each of the pathways in the collection, which provides us new targets of we need to curate this, and this is all fine, and things like this. I talked a little bit about what do we know and what information is still missing. And so there's a project that was started by Alex Pico and Anders Rieter and Christina Hansbers, which was about knowledge in published pathway figures, right? So very often when we curate a pathway, we start with a diagram in a paper, and then we add more information. Can we actually somehow generate something out of these pathway figures directly? So I haven't had time to update this data. It's now from December 2018, but it didn't change too much since then. So currently, it's probably a little bit less that are not present in any database, but the percentages are somewhat similar. So about 40% of protein coding genes were not present in any of the pathway databases. We also saw that there's still very little non-protein coding genes, right? So mRNAs, non-coding microRNAs, non-coding RNAs, and so on. There's very few of those in the pathway databases. Many protein coding genes are only present in one of the databases. So each of these databases contributes unique knowledge. But what about these 40% of genes not present? Do we really not know anything about these in molecular mechanisms level? So what we started to look at is the pathway figures. So there's so many publications out there. Can we use all of them and somehow get structured knowledge out of these figures? So instead of doing text mining, we want to do figure mining here. And they did some initial assessment of what kind of figures we have. And there's a variety of difficulty levels of pathway figures. So about 40% are actually figures that are kind of easy for humans and easy for computers. There's 50 that are easy for humans, but they're difficult for computers because they have like kind of not the same shapes and maybe turned upside down, things like this. Then there's a couple that are easy for a computer, but hard for a human. And there's also some that are difficult for computers and humans to understand. So we thought if we focus on these 40%, can't we get something out of these pathway figures? And so we published this in 2020, where we looked at the pathway figures of the last 25 years. We did a PubMed Central image search. We got 235,000 figures. We did some machine learning to classify the figures. So to actually see, is this a pathway figure or not? Or does it just mention pathway in the type, in the capture or things like this? And we ended up with about 65,000 actual pathway figures. And these pathway figures then allow us to use optical character recognition, so OCR, to identify gene names. So we have a gene name vocabulary, and we identify gene names in these pathway figures. We're still working on extending that to metabolites, but metabolites names are a little bit more difficult. And this way we can prioritize gene sets that are interesting and relevant. They provided an interactive query tool where you can look at the pathway figures and the genes that are in these figures and so on. And we're working on creating gene sets and maybe pathway figures out of some of these pathway figures. So this is to increase the coverage of genes that we have. Now, very shortly, I'm going to show a study where we looked at a disease, Rett syndrome. That's a rare disease. Where we did some transcriptomics data analysis where we used the RDF to create a pathway-based network where we basically connect all the interactions independent of the pathway boundaries. So very often we keep the pathway boundaries, but they're kind of arbitrary, right? We define where the boundaries is, but usually that in a real situation, that actually just continues. And so we built this large pathway-based network with 27,410 unique interactions. On average, a pathway has about 35 interactions, but only a few occur multiple times, so about 3,600. There was a couple of interactions, for example, the complex binding of the 3 subunits of the IK beta kinase complex that occurs in 25 pathways. So some of them occur multiple times. And so we had this large network. And we used the transcriptomics data set from brain tissue, where they had frontal and temporal cortex data from Rett patients and controls to see which pathways are affected and which regions in this large network are actually affected. So we can do this pathway enrichment analysis again and visualize the pathways as we did before. So we can see the differences between the two brain regions, but we can do an active module analysis as well. So we have this large network and we look for a module in this large network that shares a similar expression changes. And there's, for example, a Cytoscape app called Active Modules, but there's a lot of other tools as well that does that, where you get these modules that are affected the same way. And what we saw here is that there was a lot of interactions from transcription translation processes, cell cycle and immune response related processes showing up in these top ranked modules. My last very short section is about pathway data in Wikidata and Scholia. So we really want to disseminate the data that we have as much and as far as we can. And so Andra and a group of people around Andra was leading that effort, made a pathway bot for Wikidata, where basically items for biological pathways were generated. So it stores the name, identifier, organism, the list of the components, the contributors, and so on in Wikidata to allow semantic exploration and integration of the data. So this is an example where there's a complement activation pathway. You see in which taxon it's found, the WikiPathways identifier, but also the exact match. So all structured data, including all the genes and metabolites that are in the pathway structurally in Wikidata. We also did this for identifier mapping, right? So when we started with SARS-CoV-2, we didn't have SARS-CoV-2 protein identifiers because they didn't exist yet. So we used Wikidata to add information about these coronaviruses and create identifier mapping databases based on that. So when you now click on a protein, a virus protein in WikiPathways, you actually get link outs to UniProt, to Entregene, and so on. And we're also working strongly to improve the connection to the complex portal to be able to also annotate these complexes with proper identifiers in the pathways. Lastly, the Scholia effort is to provide a visual representation of the Wikidata and kind of really exploration of the Wikidata data. And this is where we also integrated the visual representation, so the viewer of the pathways. So you don't just get the description and the title, but you also get the view, the clickable viewer of the pathway in Scholia. And what you, there's one slide gone. What you can also do is when you click on the virus proteins, you can get additional information about this virus protein, where these elements have been published before, and so on. So now I hope that this, so a couple of conclusions. So the pathway models are essential for processing our understanding of molecular mechanisms. We need to know what's actually happening on a molecular level. The different formats and representations are useful for different purposes. And it's important to realize that the more variety, or the easier the automation into these different formats works, the easier it can be reused. I think ontologies are really required to add more context and semantics to the pathways. And that's something we really saw also now with COVID. We wanted to add this evidence, but it was just really difficult. And then lastly, usually pathways try to stay small and focused, right? We don't want to overdo all the elements and make it really difficult to understand. And so having the RDF representations allows us to extend and integrate additional knowledge without adding it to the diagram directly. So it allows us to do that in an automated way outside of the diagram itself. There's a lot of people I need to thank, partly from the Maastricht University team, very clearly working on the COVID-19 and the WikiPathways project, also the COVID-19 Disease Map community, many people involved there, and the WikiPathways community as well, and all the curators and developers around the world. And thank you for your attention. And I'm happy to take any questions if there's time.
Hi, Tina. Thank you for a great talk. Before I have a question related to the Heidelberg. Let's make the Heidelberg the thread of the session. But there is a question in Discord. Maybe you can answer that first. And the question is if the pathways itself is expressed as an ontology, and how does it relate to BioPax, SBML, OpenBell, or question mark?
Yeah, so in WikiPathways, we don't have a hierarchy of the pathways themselves. So Reactome, for example, has a very clear hierarchy of the pathways. We don't have that. What we use is the pathway ontology to kind of give an ontological description of what the pathway is about. We actually often interact with the team of the pathway ontology to request new terms or things like this if we don't have one. We do have an export to BioPax. So BioPax is a semantic version of pathway information as well. When we started, there was absolutely no information about the layout that we could store in BioPax. And that was something that was for us really important. We wanted to keep the information about the visual aspects as well. We do have mappings between our RDF and, for example, how we would model it and how BioPax models it. And there is an exporter, although I think there's still, it's work in progress, let's say. So that's something we can definitely still improve and we're working on. But yeah, that was the main reason why we didn't go with only BioPax, because we wanted to have this visual information stored there. But we're regularly joining the combined meetings and try to align to these other standards as much as possible.
It's a follow-up question that eats into my question, but I will raise the question and then we can continue the discussion in the Discord channel. So Andrea continues as, don't you risk that the more you integrate pathway in a whole network, the more you actually amplify noise?
So I think that what with pathways is the situation is that there is a lot of parts of pathways that occur in multiple pathways, right? So the boundaries are just not clear. And I think that the mechanisms that we describe in the pathways, they're actually quite detailed and curated knowledge. So I don't think that you add a lot of noise. I think you actually remove overlap that way. So that was basically the aim here. We wanted to study crosstalk. We wanted to see where is the overlap. And in this one module, which wasn't very big in the Rett syndrome one, for example, we had interactions from 51 pathways, which means that the elements of the pathways are in a lot of functions. Genes have lots of functions. And I think that using that from a network perspective, you need to know what you're looking at when you interpret it, right? But I think that if you use only the pathway knowledge, then you really look at manually curated knowledge. And I think then you don't. If you start adding more protein-protein interactions, transcription factor, target interactions, and things like this, then you might need to be careful, I think. Yeah.